Ever wondered what makes viruses like Ebola, influenza, and SARS-CoV-2 so deadly and hard to combat? These invisible enemies have the power to bring the world to a standstill. Understanding them is akin to unlocking Pandora's box of medical mysteries. Today, we're delving deep into the microscopic world of three potent viruses: Ebola, a deadly hemorrhagic fever; influenza, a common yet potentially lethal illness; and SARS-CoV-2, the culprit behind the recent pandemic. Dive into the microscopic world of these viruses and unravel their secrets. Imagine the year 1976. A mysterious deadly disease surfaces in the Democratic Republic of the Congo, the Ebola virus. Ebola, a filamentous virus with a single-stranded RNA, is transmitted to people from wild animals and spreads in the human population through human-to-human transmission. It's a swift and ruthless invader, causing severe fever, muscle pain, weakness, and intense diarrhea, often leading to death. Prevention and containment are vital. Personal protective equipment, safe burial practices, and community engagement are key to controlling outbreaks. Treatment focuses on rehydration and alleviating symptoms, while vaccines and antiviral drugs are in development. Ebola was first identified near the Ebola River in 1976 during two simultaneous outbreaks. The discovery was a turning point, igniting global efforts to understand and combat this deadly disease. Despite its deadly nature, understanding Ebola has led to significant advancements in its control and treatment. Influenza, a common adversary, yet capable of causing global pandemics. This formidable foe is a RNA virus, enveloped in a protein coat that can mutate, enabling it to jump from animals to humans. It's transmitted through droplets when an infected person coughs or sneezes, or by touching a contaminated surface. The influenza virus is notorious for its ability to cause pandemics. This is due to its knack for changing its genetic makeup. New strains arise from two key processes: antigenic drift and shift. Antigenic drift refers to small, gradual changes that happen over time as the virus replicates. These slight genetic tweaks can accumulate, leading to a new strain that our immune system may not fully recognize. Antigenic shift, on the other hand, is a major abrupt change where two different strains of the virus combine to form a novel subtype. This can lead to a pandemic, as our immune systems have little to no defense against this new strain. Symptoms of influenza include fever, cough, sore throat, and body aches. Prevention strategies include good hygiene practices, such as hand washing and the annual flu vaccine. Antiviral drugs are available for treatment, but the best defense is prevention. Influenza, a virus that constantly evolves, challenges us to stay one step ahead in prevention and treatment. As we unlock more of its secrets, we continue to improve our strategies to combat this ever-changing adversary. In 2019, a new enemy emerged: SARS-CoV-2, the virus that causes COVID-19. SARS-CoV-2 is a type of coronavirus, a family of viruses enveloped in a lipid bilayer, which is studded with spike proteins. These spike proteins, in a way, are the virus's keys to our body's cells. They latch onto receptors on human cells, tricking the cell into inviting the virus in. Once inside, the virus hijacks the cell's machinery to replicate, creating more viruses to continue the cycle. The symptoms of COVID-19 can be mild to severe, with some people remaining asymptomatic. Common symptoms include fever, cough, and difficulty breathing. In severe cases, it can lead to pneumonia, acute respiratory distress syndrome, and death. Identifying these symptoms early is crucial. Prevention is based on breaking the cycle of transmission. This means maintaining good hand hygiene, wearing masks, and practicing social distancing. Vaccination also plays a vital role in prevention by training our immune system to recognize and fight the virus. Treatment for COVID-19 primarily focuses on managing symptoms and supporting the body's immune response. Antiviral drugs, steroids, and other treatments are being used and studied. But remember, the best treatment is prevention. Diagnosing SARS-CoV-2 involves detecting the presence of the virus in the body. The most common method is the PCR test, which identifies the virus's genetic material in a sample taken from the patient. Rapid antigen tests, which detect specific proteins from the virus, are also used. These tests are crucial in controlling the spread of the virus, as they help identify infected individuals, even those without symptoms. The fight against SARS-CoV-2 is ongoing, but with knowledge and understanding, we can hope to gain the upper hand. We've learned so much about this virus in a relatively short time, and this knowledge is our most powerful weapon. As we continue to study SARS-CoV-2, we become better equipped to manage, control, and ultimately defeat it. Viruses, tiny organisms with a big impact. We've journeyed through the enigmatic world of Ebola, influenza, and SARS-CoV-2, unraveling their structures, transmissions, symptoms, and methods of control. We've delved into the history of Ebola's discovery and how new strains of influenza cause pandemics. We've dissected the difference between antigenic drift and shift in influenza viruses and explored the diagnosis of Ebola and SARS-CoV-2 infections. Understanding these minuscule yet mighty organisms helps us combat them effectively. Remember, knowledge is power. Stay informed, stay safe.